Welcome, everyone. Today is November 15th. I'm your host for today's webinar, Mike Wasecki. Our topic today is identifying nanotechnology, nanobiotechnology-based solutions for opportunities in personalized healthcare. I'm going to introduce our presenters in just a moment, but let me first acknowledge that this webinar is hosted by ATE Central. You can see their icon in the lower right of your screen. ATE Central is the information hub for the National Science Foundation's Advanced Technological Education community. You can find out more about all of the webinars offered and other resources by going to atecentral.net. This webinar is supported in part by the National Science Foundation, and I should remind you that the views and opinions expressed in this webinar are not necessarily those of the NSF. So thank you. Also joining us today, before I introduce our presenters, I'd like to welcome Trevor Thornton, professor at Arizona State University and the director of the Nanotechnology Coordinated Infrastructure Southwest Center at ASU. Welcome, Trevor. Would you like to come on, please, and say hello to everyone, and then we'll go ahead and introduce our presenters.
Very good, Mike. Thanks a lot. Yes, I've been looking forward to this webinar for a few weeks now because there's so much potential in the meeting between nanotechnology and biotechnology. So I think this is going to be an exciting talk looking at the future.
Thank you, Trevor.
Where this might go.
Excellent, Trevor, and thank you. Your audio level sounds really good, so thank you for doing that. Trevor, I'm going to go ahead and change to our welcome slide to today's presenters. I'll start with Joshua Smith. He's currently the team tech lead at IBM's nanotechnology program. And Trevor, as you know, Joshua finished from Arizona State some years ago with a degree in electrical engineering and then went on to work on a variety of semiconductors like ON Semiconductor, Microchip, and he's also had experience at Medtronic. As you know, many of their things are on the medical side of things. Joshua, thank you very much for joining us today. How did you switch to the bio side, or were you always interested in it from your semiconductor days, Joshua?
You know, it's interesting. I followed more of the traditional path at the time, and when I graduated from ASU in 2004, the focus was really on Moore's Law, continuing to scale and innovate in transistor technologies. My introduction, I guess my more formal introduction to aiming these types of technologies at the healthcare sector really came here at IBM as a postdoc at IBM Research. I had the opportunity of migrating to a variety of different projects and got my first introduction with that. And my adoption as a full-time research staff member here at IBM Research came within the context, which I was very excited about, of this nanobiotechnology focus.
Excellent. You know, so many things happen at the interface between technologies, and I think that's a perfect segue. If you wouldn't mind introducing your colleague, Stacey, introduce her to us, and then I know she's going to be, you and her are going to be alternating for today's presentation. So please go ahead and introduce Stacey to the audience.
Absolutely. Okay, this is Stacey Gifford. She is the biochemist, our resident biochemist here within the nanobiotechnology group. And I'll actually turn it over to her to give her a little bit more background about herself and her expertise.
Sure. Hi, everyone. Thanks for the introduction, Josh. Yeah, so similar to Josh, I came to IBM as a postdoc, and I was also adopted as a research staff member a few years ago. And my background is in biochemistry and molecular biology. I came here after finishing my PhD at Yale University. So I have a much more traditional biology background, and we have a very interdisciplinary team of engineers and scientists working on this project.
Really cool. Thank you both for being here today and all the effort you made into doing this presentation. So go ahead and take us forward.
Okay, perfect. So the first thing we'd like to start with is addressing what the current standard is in diagnostics, particularly in cancer diagnostics. So currently today, in order to confirm a cancer diagnosis, generally most patients require a tissue biopsy, which is, as most of you know, I'm sure, the removal of tissue from a tumor and subsequent analysis using histopathology, so looking at the cell structure and cell count to understand what stage of cancer a particular patient is in and whether or not that cancer is malignant. As you can imagine, there are a number of disadvantages to tissue biopsy. Primarily, it's invasive. There are many side effects, including pain and infection. It's very time-consuming and costly. Oftentimes, there's incomplete sampling of tumor tissue, which is important when you're trying to identify specific mutations. You may miss some important aspects of that tumor, and that can lead to incorrect staging and incorrect treatment. There's often limited sample size, so frequently you're limited in the number of tests that you can perform because you only have so much sample to work with. And perhaps most importantly, this technique is generally incompatible with continuous monitoring and screening. So if you want to understand how a tumor is transitioning upon treatment with a particular drug or whether there's resistance that's happening, it's difficult to do that using biopsy. Furthermore, for some cancers, a biopsy is not even really possible. So in the instance of a tumor of the brain, sometimes it's very difficult to do these types of biopsies. So the question really is there a better way to do this sort of diagnostic? And over the past few years, there has been a solution that has presented itself called liquid biopsy. And so the idea behind liquid biopsy is rather than taking a sample of a tumor, you're actually able to take a non-invasive sample such as blood or urine, and you're able to get the same types of signals or perhaps even more signals that would indicate the stage of a disease and its response to treatment and those sorts of things. So the general idea is that for most, I'm going to bring up the, okay, I'm not seeing the pointer for some reason, but in any case, for most patients,
we're seeing here, Stacey. We're seeing the pointer.
Oh, there it is. Okay, it just
there you go.
flew in.
There it is.
Okay,
Thank you.
Perfect. Thanks. Okay, so for most patients, they go to the doctor for diagnosis once they're symptomatic. So they have either a lump or they have an abnormal blood test, and this leads to biopsy and diagnosis. But generally speaking, this can occur further along in disease progression. So even in some of the best case scenarios, you're already well into disease progression, and this correlates with a low survival rate. So the idea behind liquid biopsy in part is that if there's a molecular signal that you can use to predict disease progression before you're symptomatic, and this can be done non-invasively, so that means it can be done frequently while a patient is healthy because it's cheap, it's non-invasive, it's very easy to do routinely, the diagnostics could pick that up before the patient is symptomatic and while there's a very high survival rate. And so the advantages of liquid biopsy, beyond being non-invasive or minimally invasive, are that there's low risk and very few side effects. It's also much lower cost, generally speaking. It enables early screening and detection, but it also enables continuous monitoring. So you have the ability to perform liquid biopsy throughout treatment of a disease in order to understand how it's responding to that treatment. Generally, it requires small volumes. It can offer more complete sampling of many tissues because you get mixing of signals in a single body fluid, such as blood. You can sample from many different types of body fluids, including blood, urine, saliva, and so on, which can be selected based on the disease that you're looking at. And finally, it allows you to monitor health and disease beyond cancer. So I'll give many examples in the subsequent slides of diseases that can use liquid biopsy aside from cancer. Some of the things that are detected within a liquid biopsy sample are shown here. These are 5 classes of commonly used biomarkers. So circulating tumor cells, nucleic acids, particularly cell-free circulating tumor DNA, exosomes, which we'll focus on primarily in our talk today, as well as viruses, proteins, and small molecules as well. Some of the downstream analysis that happens after isolation of these biomarkers from a liquid biopsy sample include PCR sequencing, which allow clinicians to detect mutations that may be relevant to disease treatment and disease prognosis, mass spectrometry, which allows detection of specific proteins, as well as immunoassay, which is another targeted approach for protein detection. So I just want to go through some of the common liquid biopsies that are currently used in the clinic. And the first one that really came into the field was circulating tumor cells as a target for understanding cancer progression. So circulating tumor cells are formed from a primary tumor, which releases single cells into the bloodstream. These cells migrate into the bloodstream and then flow to other sites where they eventually form metastases. And the idea behind capturing circulating tumor cells for liquid biopsy is that these migrating cells carry with them significant information on the primary tumor cell, which allows clinicians to better understand the nature of that particular tumor. So they carry RNA and DNA, which are important for understanding mutations, as well as gene expression data, protein expression, as well as protein modification. And then they can also be cultured in vitro, which allows clinicians to understand things like their morphology and their invasiveness. And so the promise of circulating tumor cells actually reached the market through a product called CellSearch. And this essentially allows clinicians to count circulating tumor cells within a blood sample. And it's sensitive down to 1 cell per 7.5 milliliters of whole blood. But just to give you a feeling for what clinicians generally measure, that number tends to be in the range of 5 to 10 cells per mil for any given patient. It can be higher, but that's usually the range that they're looking at. So the sensitivity, while it's high, there's not a huge dynamic range for that. However, despite that, there is some prognostic value. So circulating tumor cells in peripheral blood are associated with decreased survival for many different types of cancer, including breast, colorectal, and prostate cancer. Unfortunately, this doesn't really translate into predictive power for drug response. So while it has prognostic value in terms of how well patients will survive, it's difficult to use that information for treatment approaches. And as a result, because of this lack of impact on patient outcome, it actually was dropped from Medicare coverage last year. So while CTCs offer a lot of promise in terms of prognostics for cancer, I think there's still some work to be done in this field to really see their value through. And as a result, there have been efforts to seek alternative biomarkers that perhaps offer more information than CTCs can provide. And one of the major fields that's really seen a huge explosion over the past few years has been circulating tumor DNA, which is a type of cell-free DNA. And perhaps some people may be surprised to learn that there is cell-free DNA found floating around in your blood outside of the cell. And this can occur through a couple of different mechanisms. So in particular for tumor cells, apoptosis and necrosis occurs quite frequently, and you have release of cellular DNA into the bloodstream from the tumor itself. But in other pathways, there's actually active secretion. And this type of biomarker is relevant actually for conditions outside of cancer. So in the case of pregnant women, the placenta will actually release fetal DNA into the mother's bloodstream, enabling neonatal testing from a blood draw. And as you can see here, there are a number of products that are on the market today centered around cell-free DNA and circulating tumor DNA. Many of these are companion diagnostics, which just means that clinicians are looking for tumor-specific mutations that can help guide which drugs will help a patient best. There's also panels of mutations, such as the Garden 360, and then there are a number of prenatal screens available as well, which again look at fetal DNA within the mother's bloodstream to help identify trisomies and aneuploidies, as well as common mutations. But the challenge with cell-free DNA is that you're really only looking at DNA mutations, and ideally what you'd like to get a handle on is not just the baseline mutations, but also the expression levels of those, which tend to be more dynamically changing in response to different disease progression states and also drug resistance. So things like RNA and protein give you a better handle on that type of thing. So the question was, are there biomarkers that offer all of these different molecules within a single shot? And that's really where our interest in extracellular vesicles came into play. So extracellular vesicles are secreted by cells in a couple of different ways. So there's different populations. Microvesicles bud off the surface of the cell. But most of the attention has probably gone to exosomes, which are formed through a two-stage invagination of multivesicular bodies, which then in turn form exosomes within them. These are then released out of the cell. And the interesting thing about exosomes is really that they carry sampling of the cytoplasm inside of them, as well as surface markers that correlate to the mother cell. And these carry DNA, RNA, proteins, as well as small molecules. And these have been shown to be intracellular communication mechanisms. And so if you can capture these, you essentially get a snapshot of the mother cell and the state of that mother cell, which makes them a very promising biomarker for a number of different diseases. So I just wanted to emphasize again why we're so interested in exosomes over some of the other also very exciting and promising biomarkers that are in the field. But I think for us, the advantages and also the challenges really played into our strengths and interests. So exosomes are abundantly available. They have broad application to a number of diseases. All cells secrete exosomes, and they're found in all bodily fluids. So they're relevant not just to cancer, but many other diseases, including autoinflammatory disorder, infection, pregnancy, and others. They're found in most body fluids, and again, they contain RNA, DNA, proteins, and other small molecules. And interestingly, they're protected from degradation because they're vesicular. But the real challenge for exosomes has been their size and difficulty in isolation. That's really explained the trend in publication over time. So exosomes were discovered as far back as the 60s. At the time, it wasn't realized that they had any role in cell-to-cell communication. Rather, they were thought of as kind of a trash can of the cell. They were thought of as a way for cells to get rid of waste, and so there wasn't much interest in them from a diagnostic standpoint until the early 2000s, really, when people started to realize that the RNA contained, first of all, there is RNA in exosomes, but also that RNA can be taken up by target cells and affect their gene expression patterns. And most excitingly, it's been shown that exosomes can target sites of future metastasis and kind of prime the field for receiving circulating tumor cells for further metastasis. So there's been a corresponding increase in the number of publications surrounding EV research. However, again, the challenge and the reason that this has been so delayed in part is due to the difficulty in isolating exosomes and extracellular vesicles. And so despite that, there are a few companies that are working on products surrounding exosome-based diagnostics, the primary one being Exosome Diagnostics, and they have a product on the market which is Medicare reimbursable called Prostate IntelliScore, which works in conjunction with standard PSA testing to improve the accuracy of early diagnostics for prostate cancer. There's another company, Kodiak Biosciences, which is focused more on the therapeutic value of exosomes, and they're interested in engineering exosomes to contain specific therapeutics that they can target to specific sites for targeted therapies for both cancer and other diseases. But again, I think the limitation in this field has been in isolation. So the primary method of isolation far and away is ultracentrifugation, which, if anybody is familiar with this method, knows that it's incredibly time-consuming. It requires generally very large volumes of sample. It requires skilled technicians, and it's really just not automatable or amenable to clinical applications. There are some other methods surrounding magnetic bead-based methods using antibodies, but these can miss populations because it requires knowledge of the surface markers on exosomes, which sometimes is poorly characterized, especially for specific diseases. There are some kit-based agents which use precipitation or adsorption, and this is in fact what Exosome Diagnostics uses in their methodology. But there are some limitations in terms of specificity and purity for these types of approaches. And then finally, filtration is also used, but this is generally for large-scale production for therapeutics. And so as a whole, the real challenge in this field for isolation has been difficulty in subfractionation of exosomes, high contamination, and generally the contamination is poorly characterized. Many of these methods lack reproducibility and standardization, in particular ultracentrifugation. None of these methods are well automated, and many of them require sophisticated lab equipment and very long run times. And so addressing all of these is really important in order to translate exosome-based diagnostics to the clinic. And so with that, I'll hand off to Josh, and he'll start to introduce some of the technology that we've been developing to address this.
Okay.
Joshua, before we start, I wonder if could we pause for a question for a moment?
Absolutely.
Trevor, would you come on? You've made a couple of comments in the chat window. I wonder if you could bring those forward to Stacey, because I think they're very relevant. Go ahead, Trevor.
Sure, Stacey. So it seems like the free DNA and the CTC detection is already quite well developed, but it seems to me, I think you said this, that by the time you're looking for circulating tumor cells, it's already a little too late, really, because the risk of it metastasizing is increased, I would imagine. Would you like to comment about that?
Mm-hmm. Yeah, so that's exactly right. Yeah, so one of the disadvantages of circulating tumor cells as a biomarker is that they're really not amenable to early detection because, as you said, by the time you have CTCs in your bloodstream, you're already entering into a stage of metastasis. And so, so that's one drawback, and that's why you've seen with CellSearch that they are primarily used as a prognostic tool rather than a diagnostic tool. I think that CellSearch has been a pioneer in the field in terms of enabling liquid biopsy and, you know, getting FDA approval and getting it out to market. But I think in terms of defining, you know, things being well developed, really all they're doing is counting cells. So I think there's so much more that could be done with circulating tumor cells that a lot of researchers are doing, but it just hasn't seen its way to market yet. So I think there's a lot to be seen as far as the CTC technology goes. And I think circulating cell-free DNA has come a lot further because there's a very clear technique for analysis there. And I think, especially with the companion diagnostics, you're generally looking for one mutation and whether or not it's present. And so I think, you know, I think that the application there has been very straightforward, and that's why you've seen a lot more development in that field. But I think, you know, these are both still early stages. I think we'll see a lot more from them in the future.
Very good. Yeah, thank you.
You know, Stacey, one of our participants was really convinced by your slide on liquid biopsies and wondered why aren't oral biopsies done by liquid biopsy versus tissue biopsy? It seems so good.
Why aren't all?
Yeah, it seems so good. All the advantages were stunning. Yeah.
Yeah, so I mean, so I think part of the challenge is that we need to, you need to be able to detect the same, with the same confidence levels, the same sensitivity, and the same false positive and false negative rates as a standard tissue biopsy, right? So actually proving that out takes time. So tissue biopsy has been around for a very long time. It's a very well established method for diagnostics. And so I think the challenge of the liquid biopsy field is to give care providers the same confidence in liquid biopsy as they currently have in tissue biopsy, and that's something that will take time.
Okay, that makes sense. Good. One last question before we turn back to Joshua. You mentioned that it's available in all body fluids. Do you mean things like sweat and saliva as well?
For exosomes, yes. So exosomes are present in sweat, saliva, breast milk, interstitial fluid, urine,
right.
Blood. Pretty much any sample that anyone's looked at, they found exosomes there. Cell-free DNA is a bit more limited to blood, but people have reported some in urine as well.
Okay, good. Well, thank you for that pause and questions. Something on one of your slides really caught my attention when you started talking about the size of the exosomes, and there was the word nanometer after them. So I think that really sets us up for the next thing that Joshua is going to tell us about. Go ahead then, Joshua.
Absolutely. Well, thank you, Stacey, for that introduction and the background on and technologies for these different biomarkers in liquid biopsy. I think so. One of the focuses that we have here, and it surprises a lot of people when we say that we're working on these types of things at IBM Research, because traditionally, as many of the people here, or perhaps all of them know, IBM has excelled in its hardware development for micro and nanoelectronics. And as part of that, what we have here at T.J. Watson Research Center specifically is a tremendous amount of resources, both in the fabrication facility that we have, which is the Microelectronics Research Laboratory, or MRL, but more specifically in the staff with a very long history of high-impact research that has translated to different technologies. The implementation of copper interconnects, the translation of transistor technologies or CMOS technologies to silicon on insulator or SOI are two notable examples. And I think when we talk about research at IBM at a hardware level, I think we tend to think about the core technologies such as transistor technologies, memory, and integration of these devices. More recently, IBM has focused on new computing strategies, which include, probably as a very standout example, its focus on quantum computing, certainly neuromorphic. This also includes, however, IoT platforms, and of course, what we're working on is nanobiotechnology. How do we bring all of these resources and this knowledge, bring that to bear into the area of healthcare? And and so one long-term goal for our group, as well as many other research groups and companies, of course, is to create very accessible and personalized solutions to healthcare, as Stacey alluded to. And the key to that is making the test so easy and portable that it can be done frequently to pick up the first fluctuations that may indicate that further tests or some sort of action is necessary. And what does this look like today? Well, anybody who's been to the doctor's office recently, typically you may note that there's all these boxes that sit outside the door with company names like Quest Diagnostics or LabCorp, and the idea is that samples are taken, put into these boxes, and they're shipped off to a facility where a diagnosis or panel of assays are conducted in order to assess the health of that person. And normally you wouldn't get a response back for a day, 2 days at least, sometimes a week. Ideally, of course, I think we want to move to is a situation where we're not just testing ourselves once a year if we're being good about really following up on our health, but much more regularly to catch, again, these first signs of disease. And the way that we're looking at this problem is breaking it down into a set of tiered goals in research, clinical, and at the consumer levels. The idea here is to first approach trying to pull out that proverbial needle in a haystack, that biomarker that you care about, which in our case is exosomes, but also this technology we'll describe is applicable to DNA. Some of these nanoscale biomarkers, how do you, with high fidelity, isolate that from all of the other material in a complex biological sample? And how do you automate that to go beyond the traditional way that this has been, these markers have been isolated? Adding to that, of course, you want to add detection capabilities that allow you to stream data so that you're getting a diagnosis at the clinic while you're there. And that technology, of course, could then be used to collect molecular data on new sets of biomarkers, ultimately informing technology that can be used at the consumer level so that individuals can regularly monitor themselves. And I think all of this is very nicely captured in summed up in a brief video presentation showing these three-tiered goals. And with that, as I transition to this next slide, I'll have Mike cue that video if you're able to do that.
I will, Joshua, and thank you. Go ahead and click forward one slide for me, Joshua.
Okay.
So colleagues, let me pause for a moment and tell you about this video of what we're going to do. It's going to be a little bit different from every person's machine. So when I launch the video, it's going to open in a brand new browser window on your machine. So you'll watch the video on a separate browser. It'll come through your own speakers. And then when the video's over, come back to this browser window so that you can continue watching the webinar today. So again, to repeat, this opens in a new window. Once it's done, make sure you close that window and come back to this. Stacey and Joshua, make sure you go on to mute here so we don't get feedback into our system. So I see you're muted now, Stacey. Okay, so here we go, folks. Let's see if this works. I mean, it's going to work. Just give a little patience as it gets going.
Okay.
The future waits for no one, so we refuse to wait for it. Today, that'll...
There we go.
Okay.
Sorry about that.
So I think we're back.
That's okay. Yeah, we're back.
Right,
Actually,
we
that
had
played very well on all of our test machines. So good. Thank you.
there was thank you. We had a follow-up video there. It was playing after that one, but I think what that shows, hopefully, it gives you some glimpse of the technology that we'll be discussing here and its relevance to the biology. Now, the video basically shows the concept for that long-term solution I discussed. However, when we began this group and this project back in 2014, our goal was really aimed at our aim was really at that first goal of on-chip EV isolation to allow us to extract meaningful clinically relevant samples or sorted clinically relevant samples. Let me say that a different way, to allow us to isolate EVs from a complex biological mixture. And of course, we're not the only group focused on this challenge, and there has been, as this timeline shows, an increased number of publications in this area, utilizing a variety of techniques, everything from size-based approaches such as filtration and deterministic lateral displacement, immunoaffinity approaches, as well as the use of dynamic forces such as acoustics or electric fields to separate nano-sized particles. Now, the technology that we chose to focus on, and that you'll be hearing about, which you heard a little bit about in that video, is a technology called deterministic lateral displacement. One of the nice things about starting in 2014 is there's literally tens of thousands of papers out there on lab-on-a-chip microfluidics. And so this allowed us to pick from a technology that we felt we could really refine and use them to separate out EVs. The one that we chose was, again, deterministic lateral displacement, which was developed by a collaborator of ours and friend, Robert Austin at Princeton University. And essentially how this works, as described in the video, is that you have you have a more complex particle mixture coming in at the input, which is shown at the top of this top-down diagram of an asymmetric array that is asymmetric with respect to the direction of fluid flow. And let me see if I can get my pointer here. There we go. So the idea here is that both the angle of asymmetry as well as the gap between the pillars define something called the critical diameter, below which smaller particles move in a zigzag type motion around the pillars in the general direction or in the net direction of the direction of fluid flow, whereas larger particles that cannot fit within that single fluid stream are deflected or bumped, quote unquote, along the direction of asymmetry. And we like this technology for a number of reasons. One, it operates in continuous flow, which is in contrast to common techniques such as size exclusion chromatography, which is time resolved, so you can continually separate. But it can also enrich or concentrate a sample, in contrast to the normal dilution you get from many traditional techniques. This is done by introducing a sample across the entire width of the array, and the particles that bump focus and concentrate at the end of the array and come out at that output, which can be taken in a separate microchannel from the smaller material. Additionally, you can purify. You can introduce a sample with buffer on either side streaming into the array. In this way, you enact an actual separation. The material that's bumped moves into a clean background. Additionally, you can size separate. You can use stage separation to subfractionate particles of different sizes, which has been found to be meaningful for EVs. I think there's 2 studies that recently came out this last year on the utility of that. And again, this is something that we didn't develop the DLD at its fundamental level. This is something that had been demonstrated and applied to biology, at the micron scale of cells. And these are 3 examples demonstrating these type of applications. Here we have larger circulating tumor cells being separated from the smaller red, white blood cells, as well as platelets. Applied in a staged manner, it was used to actually take blood apart that being they're separating out the red and white blood cells from each other, as well as separating out the platelets. And it was also used to label platelets, as shown here, as well as lyse cells, and extract the DNA contents from them. But what we really wanted to do was test what the scalability was, and whether or not that the physics would hold at the nanoscale. And this was a big unknown at the time. In fact, when we approached Bob Austin about this, the predominant theories in microfluidics demonstrated that by calculation that diffusion would overwhelm the separation process if you took it to this scale. What we found out experimentally, however, was that this is not the case. There are subtle modifications to the process, but diffusion can be controlled by running the sample fast enough through the array. And this is what's happening here. This darker contrast region here is the nanoscale DLD array. And of course, you cannot see the pillars in an optical microscope image. But the idea here is that we're running sample in from the top, and we're jacketing that with 2 side buffers to use this in a purification mode, applied to polystyrene beads. In this case, it's a bimodal distribution of 50 and 100 nanometer polystyrene beads. Each of those particles, as it hits the array, is interrogated by the system. And the net is that we have the 50 nanometer beads moving directly in the direction of fluid flow, whereas the 110 nanometers beads come out up against a collection wall at the output. And of course, you can extract those two those compartmentalized separated particles, one from each other at that output. We also found that it can be used to separate not only spherical colloids such as exosomes, but also polymers such as DNA, which is being shown here. I'm not going to go too much into that. Again, I'll keep the focus on exosomes, which has been one of the primary focuses of our group. What we found is that we could actually separate round particles such as exosomes down to about 20 nanometers, which captures the entire range of EVs. Additionally, we found that if you have a more polydispersed sample, such as exosomes, you don't just get a bimodal separation, you actually get a splayation, a prismatic splitting of those contents at the end of the array, which allows you to subfractionate. But we wanted to better understand the partial displacement modes that we're seeing here. So this prismatic splitting, again, indicates that we have states that exist between zigzag and bump. So moving to the next slide, I won't dwell on this too long. This is a paper for those who are interested in describing the physics of what's happening here at the nanoscale. I'll be talking more about the utility of it, but essentially we modeled this. This was a work done by Sung-Chul Kim, another member of our group who handles modeling. It models the array using a unit cell, and it allows you to control basically 3 parameters here: the diameter of the pillars, and the pitch in the dx and dy direction. And what this plot is showing, and what these fluorescent microscope images are showing, is that the ratio of the pillar diameter to the pitch in dy really controls this partial displacement mode. 0 here represents a perfect zigzag condition, 1 represents a perfect bump mode condition, and partial displacement is everything in between. And here you can see where we're feeding the array sample across the entire array. These are just, again, polystyrene beads. We're able to partially deflect these populations at the eggs of the array by different amounts. So why is all of this interesting? Well, it's the new utility that this understanding allows, I think, that's perhaps most interesting. So if we look at that utility, one of the things we've been able to do from this is that if you actually have an array system placed in series with a nano-DLD array downstream, we can create what we call a condenser device. And this device has the same gap size as the nano-DLD array the downstream nano-DLD array has, but it has a larger pitch and larger pillars, which is more agnostic to particle size. This allows you to push more or less most or all of the particles up against a wall, and then you get full separation. So here you have a full width injection mode device that is allowing you to separate or completely purify particles downstream. This can also be implemented into more exotic multi-stage separation implementations. And here we've got a condenser which is focusing a several different size fragments of DNA, and you can bend the different particle size fragments around these arrays, bending around almost like an optical system or a lens would bend light. So this allows us then to, at the end here of this array, separate out 3 different fragments of DNA, while introducing all of these fragment sizes across the entire width at the inlet. One of the things that I've focused on, I guess, up till now really is the capability of what nano-DLD can do to separate out nanoscale particles. However, the primary issue with separating something down, especially a technology that's separating particles from with nanoscale features, is that you pay a big price in throughput or the fluid rates through the device as you do that. And you can see here that this throughput for these prototype devices demonstrated in literature so far are 0.2 microliters per hour at 10 bar, and pressure is the way that we drive the fluid onto and through these chips. It turns out the way to really overcome this, you have to parallelize many arrays on a single chip. And in doing this, we've been able to create chips that have over 1,000 arrays, as well as 7,600 arrays in what we were terming here large-scale integrated designs, borrowing from the microelectronic definition. And here we've been able to get at pressures of 10 bar, go from 0.2 microliters all the way up to about 900 microliters an hour. And certainly what we're calling lightly and moderately scaled are something that we're adding on to and really pushing the limits of how many arrays we can fit on a single chip. This particular slide really, I don't need to go through this too much. This is actually in a paper that was recently accepted as of today, which will be available in Lab on a Chip shortly. But the idea here is that we've created a process flow that lends itself to wafer level manufacturing. However, the final device shown here in step 5 is actually a very non-conventional type of device that you'd process. It has essentially thin silicon on glass. The glass here is about 700 microns. The silicon is about 100 microns thick, or roughly the thickness of a sheet of copy paper. And that's necessary in order to introduce fluid onto these devices without having to use any type of structured glass. The fluids are introduced and extracted from the backside of the chip. So advancing on, what I'll say about this slide, this really talks about the operation and how this structure works. In order to make this work, it was a bit of a challenge in getting this to function properly. What that we're showing here with these red arrows is sample how it's being introduced onto the chip. Its flows sample along a common bus network. Then these darker regions are actually rows of arrays. And the bump material, the material that isolates the EVs or larger particles that you want, goes into another separate common bus that is indicated by these blue arrows and out the back of the chip. The challenge here was to what do you do with all the smaller material that doesn't bump? The problem maybe could be analogous to considering 2 types of cars, bumper to bumper and only one-way traffic that feeds a residential neighborhood, which would be given by the arrays here, where each of those sets of cars are flowing into every house. Let's say you have a blue, a green, and a purple car, and they're going in and they get separated at the house and they drive out to the back alley, but you want to maintain or preserve that separation. Well, that's impossible to do if everything is bumper to bumper traffic. And so unless you create a tunnel for one of those cars to go out in the backyard where it can kind of go underneath. In other words, you need multi-layered or a 3D arrangement. And we accomplish this by introducing a set of localized vias at the end of each array pair. And so that smaller material actually drains out into these localized vias. And that's what you're seeing here in this chip. If you backlight that, you have hundreds, on some chips, thousands of these via holes that can corral into a common reservoir on the backside of the chip and be collected. As far as the actual operation of the device, what we're seeing here again, is we have twinned or paired arrays. So the angle of asymmetry for one array is going in one direction. Its twin array on the other side is a mirror image of that. So they push the bump material to a common exit point, and that exits into to bump microchannels and into a common bump outlet bus. And here you can see in operation with our fluorescence microscope image, you've got actually streams of these larger particles emptying into that common bus network. The net of this is that these devices actually preserve the displacement behavior that we see in the single array devices and allows us now to get clinically relevant volumes off of these chips. The last thing that I'll mention here is our focus to, in addition to just designing chips, we want this to be a usable automated interface. And in order to do that, what most groups will do is if they have a lab on a chip device, they put that into a flow cell, and you normally see all sorts of tubing that is allowing them to introduce and extract sample from the device. Here we wanted to completely decouple the chip from the actual system that's running it. And we do that by creating a cartridge that's run based on air pressure. Now, this is a prototype version. We're working on a commercialized version of this in conjunction with a partner. And the idea here is that you simply pipette a input sample of urine or serum into the input reservoir. You place that into our benchtop prototype system, and air pressure pushes down on that fluid surface like a piston, driving it through the chip. And so if we start here, we'll use polystyrene beads. You can see we've got a very densely populated 100 nanometer polystyrene beads in the input reservoir. As we apply that, it actually gets separated, and you end up with a clear fluid here and all the bumped concentrated particles in the bump reservoir. And so this is sort of our vision of how we get this to create an automated benchtop push button system to do this isolation rather than using the conventional EV isolation techniques that Stacey described. And with that, I'll go ahead and turn the time. I don't know if we have time for questions or if there are any questions, but if not, I'll turn that time back over to describe how this technology has enabled EV isolation and some of the results there.
Joshua, let's pause for a minute. Trevor, did you have a comment or anything you'd like to add here?
No, I'll hand over. There are a couple of comments. Dr. Saha mentions quantum thermodynamics and quantum biology. That's an interesting concept. I haven't thought too much about quantum biology.
Yeah, that question, here's the question, and it takes us off track a little bit, but the question is, do you see in the way of your research, this quantum biology is that seems to be getting a lot of press lately of people looking at quantum effects, energy, everything from things like that in biology. Do you see that as an emerging field in the area of medical diagnosis? I know it's not directly related here to the exosome research, but just as a curiosity, do you see that in the things that you've seen, Joshua or Stacey?
I can comment on that a little bit. So I think maybe that question came from the fact that there was a little bit about quantum and IBM's involvement in quantum computing. So there definitely is some interest, I would say, in the field in general. As far as I know, I'm not an expert on quantum computing by any standards, but I think some of the initial applications that people have really flagged for quantum computing do have to do with quantum biology and also modeling chemical reactions. And I think that, especially from the maybe not diagnostics necessarily, but from the pharmaceutical side in particular, I think being able to accurately model the types of reactions and the types of chemistries that are being targeted with some of these drugs is a very hot area for early applications of quantum computing. So I think, I don't know necessarily for diagnostics, but I think, as we get further along in that, I think also like encryption and security is a big area. And so maybe not necessarily on the biology itself, but on the security of diagnostics, I could see that being a big area.
Good comment. Joshua, I just wanted to make a comment of my own. I suddenly realized in that lower left slide that we're looking at, the third on the right, you're actually showing the separation of those 100 nanometer particles through this thing. There they are. I'm looking at them practically.
Yeah, that's actually taking 100 nanometer particles and not only separating them from the fluid, but actually
Yeah.
concentrating them, which again is a desirable component to have when you're dealing particularly with biological samples, but it's useful for a variety of applications
There
as
it
well.
is, right there. I'm looking at it. That's pretty cool. One other question that did come up from the audience: are these arrays that you're developing, these um, these microfluidic devices, are they reusable? Are they single use? What's the plan for them?
So if you're going to use these ultimately for clinical use, you would always run the risk, of course, the very real risk of um,
Sure.
sample-to-sample contamination from patients, which is something that you wouldn't want. So the short answer to that is yes, these would be single-use devices. The cartridge that we're developing is much thinner and has reservoirs in this. It's a plastic version. What you're seeing here is in this cube-like version is a showpiece made out of acrylic, and you actually have to mount these chips. But ultimately, you have to, because it is single-use, it has to be cheap enough to use that it makes it worth it. And certainly, I think that we can be that this technology can be made, I should say, price competitive with the existing EV isolation techniques that are on the market. But again, a lot of those, all of them are very hands-on and require a bit of training from someone trained in biochemistry or at least a technician.
Yeah, I think for research perspective, it's not something that we've spent a lot of time investigating, but there is some possibility of reusability for some of these chips. But like Josh said, since our focus has been clinical applications,
Sure.
we've mostly focused on getting them to a point where they could be disposable.
They have. So there are applications where that is not an issue. Certainly, there are the longevity of the chips. They'll survive long enough to run
Right.
more than one
Right.
sample type if the application lends itself.
Well, good. Thank you for that response to that question. Stacey, we're turning over to you, right? And I'll let you know that we've got 7 minutes to go, so give us the highlights.
Okay, great. Yep, I think
Right.
that should be fine. So Josh led us into essentially application of the technology that we've developed to this point for EV isolation from some targeted clinical samples. So when we started this off, we were primarily interested in starting with urine and serum, since those are such popular sources of diagnostic fluids. And what we used for our analysis is a method called nanoparticle tracking analysis, which essentially measures both the size and also the concentration of nanoscale particles in a particular sample. And so this allows us to measure things like yield. And so what we're showing here is that we can obtain a yield for serum and urine, total yield of about 60% if you add the two together, because we can collect zigzag and bump. But we do anticipate that based on the gap size and the targeted isolation size, most of our EVs will be present in the bump fraction, which has a yield of about 50%. We show specific isolation of EVs using TSG 101, which is an exosome-specific protein biomarker. And by ELISA, we show enrichment into the bump fraction over zigzag. And then finally, using cryo-electron microscopy, we also show that we're isolating intact extracellular vesicles, which are the correct size that would be anticipated, and we're not damaging them or lysing them by separation in the DLD device. We then wanted to benchmark our method against existing exosome isolation methods. So we looked at ultracentrifugation plus density gradient, QEV, which is a size exclusion chromatography method, ExoEasy, which is the method used by Exosome Diagnostics. It's an adsorption column, and then our method. And so for serum, we show that we have significant improvement in both the concentration factor coming out of the bump fraction as well as the total yield. This is just the bump yield, since that's where most of our EVs are found. And if we look at the yield versus processing time, we have significant improvement in both yield as well as reduction in processing time, giving us a clear advantage over some of the other methods that are out there. For urine, we found actually that ExoEasy outperformed us initially for concentration factor, although not in yield or processing time. But this allowed us to really demonstrate the versatility of the technology. So in our initial chip design, we had a bump outlet, which was approximately 1/10 the width of the full DLD array. By redesigning this
outlet to be substantially smaller, we were actually able to increase the concentration of the extracellular vesicles in the bump fraction to 60 times the initial concentration. And this is in comparison to the the fourfold increase we were seeing in our initial nano DLD chip. And this is actually 6 times more than what ExoEasy is able to obtain. So this really demonstrates the versatility and adaptability of the technology depending on the end user's need. And that actually leads to a nice transition to some of the end user applications that we've started working on. So we're collaborating with the team at Mount Sinai, and they're particularly interested in understanding how we can better differentiate aggressive versus indolent prostate cancer. So in the onset of prostate cancer, you'll see that a primary tumor forms within the prostate, which can be observed under the microscope as tumor cells invading the prostal duct. And after primary tumor formation, there's 2 different pathways that can occur. The patient can either have organ-confined prostate cancer, which is essentially indolent prostate cancer, meaning that the tumor stays within the primary tumor site and does not leave or metastasize. Or the other option is an aggressive prostate cancer, which essentially leaves the prostate and can invade other organs such as the brain, lung, liver, or bone. Clearly, the outcomes for aggressive prostate cancer are much worse than those of indolent prostate cancer. Most patients who have aggressive prostate cancer will undergo a prostatectomy, which can lead to a number of really unpleasant side effects, such as incontinence, hormone deprivation, and other complications. So the goal is to be able to differentiate these populations better so that patients who have indolent prostate cancer don't get an unnecessary prostatectomy. Currently, the methods for differentiating these two are based on histopathology, which can lead to a lot of errors, is incredibly subjective. You have differences between doctors, even differences between days of analysis within the same doctor. And so you have a lot of patients who are getting prostatectomies who don't need them. So the question that we had was, can we develop an RNA-based exosome biomarker panel to help better separate these 2 populations using molecular information from a liquid biopsy? So the first thing that we wanted to do in this process was to understand what our reproducibility was from a technical level. So we performed the entire pipeline of RNA sequencing from sample collection down to RNA sequencing itself. And we looked at replicates at the exosome isolation level because that's where our technology comes into play, as well as RNA sequencing itself, which is generally considered to be the most reproducible step in the pipeline. So this is essentially the baseline of what we can expect for reproducibility. And that is shown here in this green square. So what you're seeing here is essentially these, the 2 replicates, their counts per million plotted against each other. So in a best case scenario, what you'd expect to see in a perfect replicate is that they would all line up along this red mean line. But obviously there is some standard deviation, which is reflected by the variance from this red line. But this is essentially, again, the baseline variance that we can expect from this process. When we move to variance of DLD replicates, you can see that the standard deviation is a bit larger, but it's still substantially low compared to what we see for ultracentrifugation, which has significant standard deviation from the mean. And this is better reflected in the plot here at the bottom, which shows in the green line, the standard deviation for sequencing is quite low. DLD in red is a bit higher, but not nearly as high as ultracentrifugation. So we can conclude from these data is that we offer improved reproducibility in the RNA sequencing pipeline, which is important in diagnostics in order to ensure increased sensitivity and reduce false positives and false negatives. And so finally, what we wanted to do with the technology, just as an early stage point of proof of concept, was to show that we are isolating the classes of RNA that are expected to come from exosomes, as well as specific prostate cancer biomarkers that others have published in the literature. And so what you can see is that we're able to isolate predominantly microRNA and long non-coding RNA, which are the 2 primary classes shown to be present in exosomes. And we also isolate a reduced amount of rRNA, which is generally considered to be a contaminant compared to ultracentrifugation. So we have a cleaner prep than ultracentrifugation. And then if we look at a curated list of 50 prostate cancer-related RNAs, we're able to detect all of these different biomarkers, again, as a proof of concept that compared to ultracentrifugation, we can detect the same biomarkers with less volume, with less processing time, and with greater reproducibility. And so I think this really demonstrates the applicability of DLD in the diagnostic pipeline, and this is something that we're continuing to pursue. And so just to quickly wrap up, I'll give a quick conclusion slide. So I think what we've shown you today is that EVs offer a diverse array of biomarkers for disease from RNA, DNA to protein, but isolation of EVs presents the greatest challenge for clinical applications. We have started to address this using our nano-DLD technology, which offers improved concentration, yield, and processing time, as well as increased reproducibility for the RNA sequencing pipeline. And in the future, we're continuing to work to develop this panel of RNA biomarkers in order to differentiate indolent versus aggressive disease. We're working to engineer chips which will increase purity. So our current focus has been on yield and concentration. We're working to add purity to that as well. And we're always looking for partners to collaborate with for new EV applications as well as other applications in cancer and beyond. And so with that, we'll take any additional questions. And
Excellent,
thank you guys so
Stacey.
much for your time.
You know, colleagues, we're right at the top of our hour, and so if your schedule requires you to go away, that's fine. But we're going to spend a few minutes talking with Stacey and Joshua and answering some questions. But again, if your schedule pushes you, please feel free to exit the webinar if you need to. Trevor, what do you make of this? It's pretty exciting. I tend to almost forget that this is totally enabled by nanotechnology here.
Yeah, it's fascinating, isn't it? I think it won't be too long until we're all seeing these things in our doctor's offices when we go and visit, and we'll be spitting into a lab on a chip and getting the results almost in real time. So yeah, it's amazing. As I was hoping, fascinating connection between nano and biotechnologies. There was one quick question maybe Josh could answer. So RJ was asking, how can traditional semiconductor fabs in a sort of research lab adapt to wafer level processing? So that's something I struggle with at a university. We can't produce 8-inch wafers